What if you could live for 125 years? What if we could reverse aging or even slow down its process? Before, it might have sounded like something straight out of a science fiction movie, but in actuality, the stem cells do have this potential, and there has been research done on it, an experiment done on it, that shows that perhaps you can use stem cells to, in fact, reverse aging and somehow find an anti-aging mechanism. Before I get into the experiment, how does aging exactly work? So previous research done actually shows that the hypothalamus plays a crucial role in controlling aging. The specific cells that are actually responsible for the aging process are called neural stem cells. And then researchers found out that the number of brain stem cells in the hypothalamus steadily decreases with time, and this impacts the speed at which the aging process unfolds. However, they also found that the effects of these are not irreversible, and we can regenerate them using stem cells or by the molecules produced, and it is possible to slow or even reverse the aspects of aging throughout the body. How cool is that? Now, to see what the actual correlation was between aging and hypothalamus cells, researchers actually conducted an experiment on middle-aged mice. So what is the hypothalamus? What the hypothalamus does is it's known to regulate important processes including growth, development, reproduction, and metabolism. It also helps to regulate many basic needs and behaviors in the human body, including hunger, sleep, fear, and aggression. If you ever feel yourself panicking or angry, then your hypothalamus is probably controlling this temporarily. We have also pinpointed the cells in the hypothalamus that control aging, a tiny population of adult neural stem cells, which were known to be responsible for forming new brain neurons. So what researchers did was they tested this correlation between aging and stem cells on middle-aged mice. And so at first, researchers made sure that cells with protein markers of neural stem cells, in this case SOX2 and BMI1, were present in the hypothalamus of early to middle-aged mice. Next, they destroyed the neural stem cells in the brain by injecting the third ventricle adjacent to the region where the stem cells are found. How they did this was with a lentivirus that was actually able to convert a compound into a toxin in cells expressing the stem cell marker SOX2. Then, 3 months later, the researchers looked at what this experiment resulted in, and they looked at the muscle endurance, social behaviors, different performances of the mice, and they compared the mice they injected with the virus and how they reacted. And the results were amazing. The mice in the experiment group lost 70% of their hypothalamic stem cells, and based on the results of the psychological test, had accelerated aging. However, researchers just didn't stop there. They also implanted mice with stem cells that were taken from newborn mice to see if additional stem cells could actually help in slow aging. But because of the environment of the hypothalamus, most of the stem cells actually died. But then, what they did was they genetically engineered these newborn neural stem cells to withstand this environment, and then they did survive the second time, and were able to perform better on age-related measures than the other mice. Scientists also looked instead for some factor that might be responsible for the stem cells' effects. What they found was the stem cells release microRNAs, which are a type of molecule involved in the regulation of gene expression. The microRNAs are contained by exosomes, which are particles that the stem cells release into the fluid found inside and around the brain and the spinal cord. What the researchers specifically injected into the mice were exosomes containing this that had been collected from hypothalamic stem cells. These attracted exosomes were released into the mice's fluid and allowed to act. What we can get from this information is that with further research, we can establish which particular microRNAs and other particles released by neural stem cells contribute to the rejuvenating process. So whether humans will be able to live another 120 years is actually hard to tell right now. But just the mere fact that we could potentially reverse aging with stem cells is just amazing. And if the mouse work is able to translate into humans, that means in the future, stem cells have the potential to slow down aging and perhaps even increase longevity.